In the past, it was believed that proteins were the genetic material. However, in time, through the various experiments using bacteria and viruses, the truth about genetic material came to light. British bacteriologist Frederick Griffith was the first to conduct experiments with bacteria Streptococcus pneumoniae that was responsible for causing the disease pneumonia. He worked with two types of bacteria strains, the pathogenic S strain and the non-pathogenic R strain. The S strain has a polysaccharide coat that makes it smooth, whereas the R strain lacks it, making it rough. During his experiments, Griffith realized that when heat-killed S-strain cells were mixed with R-strain living bacteria, and this mixture was injected into healthy mice, they died of pneumonia. Thereafter, when the bacteria were isolated from dead mice, they were found to be of the living S-strain type. This proved that the R-strain bacteria had transformed to the living S-strain. Yet for a long time, due to lack of conclusive proof, proteins were believed to have caused this transformation. However, in 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty worked together to determine the biochemical that had caused the transformation in Griffith's experiment. They repeated Griffith's experiments and purified all three biochemicals such as proteins, DNA and RNA from heat-killed S-strain cells to identify which one had caused the transformation. They discovered that neither the protein-digesting enzymes, that is, proteases, nor the RNA-digesting enzymes, that is, RNases, affected this transformation. Whereas DNA-digestive enzyme, DNase, did inhibit the transformation. Therefore, it was clear that the transforming substance was not protein or RNA, it was DNA. 8 years later, in 1952, the definitive evidence that DNA was a genetic material was provided by Alfred Hershey and Martha Chase. They conducted their experiments with viruses that attack bacteria, better known as bacteriophages. Bacteriophages attach to bacteria and implant their viral genetic material inside the bacterial cell. The bacteria treat this genetic material as their own and produce more of such virus particles. Since Hershey and Chase wanted to find out whether it was protein or DNA from the virus that entered the bacteria, they grew these viruses on two different mediums, one with radioactive phosphorus and the other with radioactive sulfur. As DNA contains phosphorus while protein doesn't, the viruses that were grown in the presence of radioactive phosphorus had radioactive DNA and not radioactive proteins. On a similar note, as DNA doesn't have sulfur like proteins, the viruses grown on radioactive sulfur had radioactive proteins and not radioactive DNA. Thereafter, both these radioactive phages were allowed to infect E. coli bacteria. Later, after the infection, when viral coats were removed from the bacteria, it was revealed that the bacteria that were infected with viruses containing radioactive proteins were not radioactive. On the other hand, the bacteria that were infected by viruses with radioactive DNA were radioactive. It was clear then that proteins did not enter the bacteria from the viruses. Rather, it was the DNA that passed on from the virus to the bacteria. Therefore, despite experiments by Griffith and Avery, MacLeod and McCarty, it was the experiment by Hershey and Chase that proved the fact that genetic material is DNA.